Everyone. Sorry for a little bit of a delay. We had some technical issues we had to sort out. Welcome to the Theoretical and Computational Biophysics seminar. Today we have a guest not far from here, even though remotely it doesn't make any difference. So we have David Minh from Illinois Institute of Technology, or Illinois Tech, chemistry, who will be talking for us. Before introducing the speaker, again, I remind everybody to please turn off their microphone before the seminar starts. And then again, type in your questions in the chat box, and we either call on you to ask your question during the seminar if David doesn't mind, or we actually sort of wait until the end of the seminar where we can actually ask our questions. You can keep your cameras on, so that actually is encouraged. So you have we have a more kind of interactive session. It's not like David is talking to his computer, but please turn off your microphone during the talk. So now let me give a brief introduction. David is a chemist by training, so he did his initial degree at Berkeley in 2003, and then he moved to UCSD, if I'm not mistaken, with McCaman, where he did his master's and PhD in physical chemistry, and where he started actually working on free energy problems and techniques. Then after that, after a postdoctoral period at NIH, he actually received the prestigious Director's Postdoctoral Fellowship from Argonne National Lab, where he was for a couple of years, and did another postdoc at Duke University before he joined Illinois Tech as a faculty member, as an assistant professor, and he went through the ranks, and he is now a named faculty in chemistry at Illinois Institute of Technology, at the same time Associate Director for Center for Interdisciplinary Scientific Computation. He has received a lot of awards during his student time and his postdoctoral times, and I'm not going to go through all of them. I only mentioned a very interesting award called 40 Under 40 Chicago Scientists, which is very, very nice. It's a long list of publications. So research-wise, he is one of us. He's interested in application of molecular simulation to interesting molecular problems and developing the theory, developing software actually, which is what we do here too. So I expect to have a lot of common interests. So thank you very much, David, for being here and accepting our invitation. We look forward to your seminar. Please.
Thank you, Imad. Thank you for the very nice introduction. I've also had a lot of positive experiences with UIUC and the center. Just learning about the software tools and also the workshops and just the research going on. It's been high quality work that you do there. So yeah, thank you for the invitation. Today I want to talk about one of my favorite projects to work on, or that I've been working on the last almost a decade now, which is what I call implicit ligand theory. And this theory enables you to do large scale free energy calculations, and I'll be talking about that in detail. Well, Imad mentioned that questions could be either during the seminar or after. So yeah, I'm fine with you asking it before or during the seminar. That way I can, we can have more of a discussion rather than just me presenting.
Thanks.
Okay, so the outline of this talk is I'll be talking about the definition of the standard binding free energy and what would be some of the potential applications of being able to do it quickly. I'll also describe some of the established methods and the tools and tools in the field, including molecular docking and alchemical pathway approaches. Then I'll describe this theory and where it could fit into the whole niche of binding free energy methods. I'll show you what it looks like to do a binding free energy calculation to a rigid protein that I call a binding potential mean force. I'll describe some of the key equations and then some of the example calculations that we've done with T4 lysozyme and small molecule inhibitors or binders of T4 lysozyme. Finally, I'll talk about how you might pick out snapshots for doing free energy calculations too. This is actually applicable to molecular docking as well. It's sort of an open question. And we applied a statistical methodology to help understand how to address this question. Talk about some other ways that we've been developing or some other directions that we've been taking, that we've been able to use the fast Fourier transform to estimate binding potential mean forces and also get free energies that are relatively accurate or more accurate than interaction energies. And also a different way to formulate the theory to get relative binding free energies between related compounds. And then finally, I'll mention how we can get ensemble averages of a protein bound to a small molecule when you start with an ensemble from an apo ensemble or without any ligand bound. And when you have this hollow ensemble, we can make predictions such as, is a small molecule an agonist or antagonist of a membrane receptor or nuclear hormone receptor? And a lot of this is published. Some of it's preliminary work that hasn't been published yet. And I'm welcoming your feedback on, well, published or unpublished. Okay, so first of all, I just want to introduce what is the standard binding free energy. Basically, it's a logarithm of the dissociation constant. And yeah, so basically the dissociation constant is the concentration of a receptor and a ligand, or free receptor and free ligand over the concentration of the complex at equilibrium. And then the binding free energy would just include the beta to -1, or the inverse beta times the logarithm of KD, and you divide it by standard state concentration, because, well, technically they all should be divided by standard state concentrations to be thermodynamic or approximations of the activity. And, well, you end up with one more concentration on top, so you end up with an extra standard state concentration on the bottom. There's a large range of binding free energies and dissociation constants which are observed in biology. For interaction between like ATP and a kinase, you might get something in the millimolar range. For the highest known biological affinity, you're talking about something in the picomolar range or -16 kcal per mole binding affinity. So if we can make large-scale free energy predictions, there are many applications, but I'm just going to talk about one of them. One of these is for virtual screening. So the way you can pose this challenge is that if you have a library of chemical compounds and a three-dimensional structure of a receptor, what compounds actually bind to the receptor? And if we can answer this question well, if we can answer it accurately and quickly, then this could be useful for designing chemical probes to interrogate or to study the function of these receptor molecules, these proteins, in vitro or in vivo. You can also use it to design drugs that will target those particular receptors. And then if you're talking about something like a nuclear hormone receptor, you might be able to find small molecules that bind to it. And if you're talking about an enzyme, you might be able to find substrates that are processed by the enzyme. So these are very important problems, and there are many ways to do binding free energy predictions. Here I pose them contrasting their accuracy on one axis versus their computational expense on the other. I say log because it's not really on the same scale. You have calculations that take seconds, like docking can take only seconds if you do it with very quick parameters, or parameters that are amenable to doing it quickly. And on the other end, you can just run a full all-atom molecular dynamics simulation for a long time and just see molecules bind and unbind. And in between, we have the whole gamut. You have endpoint approximations where you run simulations of a bound system and an unbound system. You also have alchemical approaches. So yeah, I'll be mentioning these in more details. This is not meant to be an exact figure by any means, but a general idea. So in any case, docking is the fastest and least accurate method. And it's been found that basically there's no correlation between the score that comes from molecular docking and a binding affinity. And this review is somewhat old, but there haven't been major advances in docking that would lead to major changes in their accuracy for binding affinity prediction. Molecular docking is often another way that you can use docking is not to actually predict the binding affinity, but say, okay, compounds that are active are going to have a lower docking score than those that are inactive against a biological target. And in this task, it's inconsistent. I mean, most of the time, well, you're going to have different programs that work well for different targets, and it's not always predictable exactly what is the best program. One thing I want to say about this particular figure is that this is a separation of active compounds from decoys. So decoys have similar chemical properties as the active compounds, but they have a different connectivity. So this is a little bit of a more challenging task than picking out active compounds from a completely random library, like the needle in the haystack, because in the haystack, you're going to have a lot of hay that's irrelevant to binding, whereas these decoys at least look like compounds that bind. But oh yeah, I didn't really discuss in great detail, but these are rock plot curves. So these curves are really good if you can go straight up and then straight across. And they indicate that you find all the actives before you find the decoys if it were going straight up and then straight across. If you're going along a diagonal, then you have a completely random classifier. And then if you went straight across and then up, then you would have a completely terrible classifier, which arguably could be perfectly good if you flipped it around. But yeah, in many cases, you have something that is pretty much random. And in other cases, you do have a pretty good classification. But yeah, you might have a large variation between the different docking programs. So as I mentioned briefly, you can also just watch a really long simulation. And this has only been done a few times because most of the time you don't get very many statistics from it. You might just see one binding event and then it's kind of boring. You might just see it stay the same. But this is a nice proof of principle about in 2011 where basically it demonstrated that the force fields were accurate enough to recapitulate the crystal structure of or co-crystal complex between the satinib and a sarkinase. But this is not something that's done routinely just because it costs a lot of computer time. The alternative that has been increasingly popular and is available in commercial packages and has really gained traction in the pharmaceutical industry are alchemical pathway calculations. Basically, the idea being instead of trying to, well, basically we have one thermodynamic state in which a receptor and ligand are bound, and are unbound, and another thermodynamic state in which they're bound. And these alchemical pathways involve a number of non-physical, non-chemical intermediates that are partly between one state and another. So yeah, there's two ways that people do this. People will either pull apart a pair of molecules with maintaining the same sort of force field, or they will morph one small molecule into another. This particular pathway is showing complete dissociation of a small molecule from the receptor through a series of fictitious states that include removing some harmonic bonds or stretches that are completely fictitious, and then adding them in different stages. This whole field is possible because of thermodynamics, and the thermodynamics involves state functions that don't depend on the pathway that you take to go from one state to another. But yeah, this is much faster than just watching it simulate, but it still takes a while, even with all the advances we have in computing and use of graphical processing units. Yeah, so these methods have become a lot more accurate than in the past, with improvements in force fields, improvements in sampling, improvements in different technologies used to estimate free energies in the last 2 decades or so. There's a seminal paper about 5 years ago that was released that involved a collaboration between the computational chemistry company Schrödinger, and they were working with 8 different pharmaceutical companies, and they made predictions about compounds that were being proposed to be tested, and then they compared these predictions against actual values after they were tested, and you can see that it's not random. Basically, there's a strong correlation, and there's a lot less error between the experimental and the predicted than with docking, for example. There are some limitations of this data set, or there were some limitations of this data set, and a newer data set came out earlier this year. This was an internal study done by Merck and just published in the open literature this year, and they found cases where there is larger error in some of the systems that they worked with. They also released some benchmarks from publicly available data using the same sort of approach. So yeah, these are showing that alchemical pathways are getting a lot better, but they still are computationally expensive. They were about 400 calculations done by Merck in this 2020 study, and then about 500 in this previous one, which is not approaching the scale of libraries that we're talking about for virtual screening. So that's why I'm going to talk about where implicit ligand theory can fit in. Before I move on, any questions?
I have a question, David. I remember the Schrödinger data set. Many people were complaining that we don't know their force field. Is that still the case? So I remember actually they were using a black box, and they said this is what we can do. So you can use the force field, but you don't know what they are, and you can't change them. That was the status I remember vaguely. I was wondering if this has changed or still the case.
I'm actually not aware of that particular criticism of the data set, but I know that there have been a number of people who've looked at the same systems and attained a similar performance using different force fields.
Okay.
So it's become like a benchmark that people are always testing their methods against. But right. There's some limitations of the benchmark in that many of the systems, they don't have large changes in free energies. I don't think they have any changes in the net charge.
I see.
Okay. And that was part of the motivation for the 2020 study, is that Schrödinger has implemented a number of new algorithmic tricks within their commercial package, and they wanted to see like, oh, did this make things better, or at least that's what I read in the papers. Like, oh, they want to see whether the state of the art has changed in the last 5 years.
Okay, thanks.
Okay, so where I see implicit ligand theory fitting into the landscape of free energy methods is as a second stage approach. Basically, you can do millions of compounds doing a fast molecular docking program, and then you can take a subset that come from this first stage, like the top thousands or few thousand, and run them with implicit ligand theory. And then as a third stage, you could either do an alchemical pathway approach, or you can go straight to the lab. A nice thing about this approach, as I'll talk about in greater detail, is that you can sort of tune a knob between accuracy and computational expense. That is, there is a clear theoretically rigorous approach to increasing your computational expense and hopefully increasing your accuracy. And it's a principled approach. And like some of the other intermediate approaches out there, there's not a clear pathway to improving the accuracy. So what I'd like to show you is what it looks like when you sample from the ensemble of different thermodynamic ensembles as you increase the strength between the receptor and the ligand. And so alpha is a parameter that describes the strength of the interaction. And at alpha equals 0, basically the small molecule is anywhere within a sphere. And then at 1, it's limited to a very small region of the configuration space. So these were calculations done using a program that I wrote, well, based on some existing software tools for molecular dynamics called AlgaeDoc. Some key features of AlgaeDoc are that the protein is considered to be completely rigid, and this allows you to do the calculation in a fairly short time relative to binding free energy calculations where the receptor is flexible, and you can do this in a sort of distributed computing network opposed to massively parallel or GPU framework. I'm also, in this calculation, also varying the temperature because you can do that when you're dealing with a very rigid protein, and that allows, that's a tool to enhance the sampling of the ligand from one binding pose to another. In this particular example, I also show the crystal structure in purple, where the carbons are in purple. So you'll see that when alpha is close to 1, the ensemble of structures is close to the crystal structure. These simulations were not done sequentially. They're done using Hamiltonian replica exchange. So they were all the different thermodynamic states were simulated in parallel. Well, parallel in a sequential way, like running a short simulation at alpha equals 0.7, another one at 0.6, and then just keep continually doing that, and then going to the next step within the Markov chain. Okay, so you might be wondering, you're talking about a completely rigid protein, a completely rigid receptor, but in solution, things are not completely rigid. What are you doing? Why am I wasting my time here in the seminar? You are not, because you can actually obtain a binding free energy, a rigorous binding free energy to a flexible protein by taking an average of the binding free energy to the rigid protein. This is a result I showed in a 2012 paper. Basically, the binding free energy can be shown, or is an exponential average of the binding potential mean force which is an exponential average of the interaction energy between the receptor and ligand. And then here, these are potential energies that include the solvation energy. So basically, R sub big R L is a configuration of the complex, a configuration of the receptor, configuration of the ligand. This is the binding PMF. It's a one-step perturbation formula that, well, here you can see that it's a one-step perturbation. This formula is a one-step perturbation, but you can actually do this using an alchemical simulation or a series of states like I do in the slide or the movie that I just showed you. And then, well, I actually do use a one-step perturbation to get the binding PMF. And this denotes that this ensemble average is over a bunch of different receptor configurations from the apo ensemble. And here's a standard state correction term. Yeah, I'm going in a little more detail than I usually do because I know you're a theorist. So I also will take a brief moment to pause for questions here.
So you do a sort of separate simulation of the protein, right?
Yes.
Protein, and just use that to get the sort of the first term that you're showing up there. And then you select one of those snapshots. Any of those snapshots could be used to calculate what you showed us in the previous slide.
Oh yeah, the previous slide would be for one snapshot, but to actually get a binding free energy accurately, you need to do this over many snapshots, or at least for more than one. So basically, makes the calculation parallel. Gotcha. And this also tells you how you get more accuracy. Basically, you can increase the number of snapshots, and at some point, the exact point is going to depend on how well you pick the snapshots and how flexible the protein is. You're going to get the same free energy as you would if you did alchemical approach with the same force field.
I see. It's not similar to ensemble docking, so to say. You're just taking many frames from your protein ensemble and then do your binding calculations on all of those frames. I see.
Yeah, exactly. So there's the same philosophy as ensemble docking, but this one actually has a foundation in theory that links it to the binding free energy.
Okay, cool. Mm-hmm.
Yeah, so it's rigorous, and this has advantages in that you can use, well, the advantage of using multiple receptor structures is that you can recycle these. So if you're doing a typical binding free energy calculation with alchemical approach, you'll need to do sampling of the receptor for every single ligand. And even if the conformations that you see from ligand A and B are the same, you'll have to see the same, well, if you have a conformational transition that happens, you're going to have to see that transition in both ligands in order to get an accurate free energy. This is also scalable. So yeah, I didn't talk in tons of detail about that movie, but the one shortcut I used within the calculation, I guess I'll talk about that soon, is that I represent the interactions between the receptor and ligand on a grid, and that allows you to compute these very quickly. And it doesn't after you calculate the grid, it doesn't depend on how large the receptor is. So you can do this on something like the ribosome.
I see.
In principle. I haven't gotten to that yet.
But yeah. So then one more question. So the simulation you showed, you don't need to know. The whole idea is that you don't need to have the binding pocket. So when you do it this way, especially if you have a great representation of the interactions with the protein, you can do it fast enough to even discover binding sites for ligands. Is that correct? Or you need to know the binding pocket?
In principle, you can do it without a pocket, but I think it's just going to be a harder search problem.
Mm-hmm.
So just like docking, you don't really need a pocket,
I see.
but you're just gonna require a longer simulation to get. So,
So
I actually do restrict it to
yeah,
like sphere for the purposes of saying, okay, the binding pocket is within this sphere.
so
But
how
it
long
doesn't
was this
have to be.
simulation that you're showing us? It's impressive actually how close it gets to the actual experimental structure. How long was this simulation to
I
get
think this one was about 2 days on a single CPU. So the length of the simulation will depend on how big the ligand is. So
I see.
and then exactly how long is required, that's also going to depend on the size of the ligand. And actually, not only the ligand, but the complexity of the binding pocket. Because if there's a lot of complexity in the pocket, then you're gonna have a little trouble going from one site to another.
Cool. Thanks.
So yeah, in this paper that I published, or it was published earlier this year, I described the performance of this approach on a set of 85 different protein ligand complexes. We're recapitulating the crystallographic pose, or starting from a crystal pose, and then how well does it recapitulate it, and how precise are the binding PMFs. Okay, so the way to apply this theory, the first step, as Imad mentioned, is to sample configurations of the receptor. And yeah, as I mentioned, this can be done once, one heroic calculation. And this is sort of the thing you might see a lot of times in the literature, that people will just try to get thorough sampling of the APO receptor. And they might do something like a Markov model of it. The second thing is to pick out snapshots of the receptor and estimate the binding free energy between the rigid receptor and the flexible ligand. And then the third is to estimate the binding free energy. Basically, you can take a weighted average of the binding potential mean force. And the weight would basically be the weight of the snapshot in the APO ensemble. So yeah, in this paper that came out earlier this year, I did this I did the binding PMF calculations using this thermodynamic cycle that involved a lot of intermediate states. All the sampling is done with these grids, and then there's sort of a bookend post-processing to get a grid-free sort of molecular mechanics energy of the samples from the end states. And there's a bunch of intermediate states. Well, the first sort of stage is like increasing the temperature of the ligand by itself. And then second is to scale in the grid. I showed you samples from this scaling in of the grid. And then, yeah, this post-processing. What I'm showing on the right is like the number of, there were 85 systems. And then you can say that, we look anywhere on this and you can say, okay, 60%, with a certain number of cycles, with a small number of cycles, you start with the blue, 60% of systems have a RMSE less than 6 KT. And then as you increase the number of cycles, then basically the simulation time, you get more and more precise. Smaller root mean square error. These are different pairs of states. So there's, this is the post-processing of the separated species. And this would be the raising the temperature of the free ligand. And this would be scaling in the grid. No, sorry, this would be, yeah, this is scaling in the grid. This is changing the, basically changing the implicit solvent model. And this is overall. And there are two ways to do this, basically with that depend on the solvent model used for the ligand. And one of these is basically saying that there is no solvent. It's always desolvated. Another is that I'm using a generalized Born solvent for the ligand. So it looks like the weakest link is trying to go from a grid representation of the potential energy to the fully solvated form. I'm using a generalized Born implicit solvent for the whole complex. Okay, so that's the theory. And I want to talk a little bit about some of our demonstrative applications. We applied this to T4 lysozyme, which you can say is the hydrogen atom of protein ligand binding. And what happens in this system is that you can do mutation of lysine to alanine and you get an artificial cavity. And when we started this study, there are 141 hydrophobic compounds, mostly hydrophobic, and they're known to be active or inactive according to a thermal shift assay. And T4 lysozyme was initially considered to be a rigid protein, but we know from crystallographic studies that there are some motions that are relevant to binding different small molecules. So the way that we did this sampling, or we actually did two ways of sampling. One of them, which is published, the other is still awaiting publication. And this work was done by my former PhD student, Bing Jie, who's now a postdoc at the NIH. So basically what she did was she did alchemical calculations with 6 proteins. And then she used snapshots from those 6 calculations to do free energy calculations using implicit ligand theory for the rest of the 141. And we can closely reproduce results from a flexible receptor. That's what Yank is. Yank is a package that is being developed in John Kadera's group. And so we get a fairly small root mean square error, pretty close reproduction of the flexible receptor results. This is for 24 different small molecules. And one thing that we noticed is that, there were these 6 different small molecules we did simulations with to draw snapshots from. And if you just pick one of them, sometimes you'll get a decent result, like with methylpyrrole, benzene, xylene, you get pretty decent results. But if you pick phenol, you would not be able to get a good correlation with the alchemical result, and your error would be very high. If you pick snapshots from the active ligands, then you get something more reliable than any individual ligand. And I think what this is telling me is that when you have different ligands, your protein explores a somewhat different configuration space. And you need to see all this configuration space if you're going to do a whole bunch of different small molecules. But I think there's a limit to how much configuration space is going to be accessed for a particular protein, even within a lot of different ligands.
What is an active protein? What do you mean by active protein? You know what is the active snapshot or confirmation?
Oh, I'm sorry. It's not an active protein. I'm talking about whether the ligand itself is biologically or active against the biological target, whether it changes the stability of T4 lysozyme in a thermal shift assay.
I see. Okay.
So we just chose, because we know that a number of these small molecules bind and cause the thermal shift to change, and others don't do anything. But we just wanted to simulate with both just to see what it looks like. And we can take snapshots from these simulations. And if we take snapshots from simulations where we know that the ligand is a biologically active ligand compound, then it's more useful than if we just take them from the ones where they're inactive. So yeah, one thing I mentioned as a niche or one key feature of using this theory or this approach to getting free energies is that you can tune the knob, increase the accuracy by increasing the number of receptor snapshots. And that's what you can see with this system. In that here we're looking at the different metrics of quality. The top two rows or the top row corresponds to accuracy with respect to yank. The bottom accuracy with respect to the final result. Like if you have all 500 something snapshots. And the different colors correspond to different ways of selecting the receptor snapshots. Red is completely random without any preference for one snapshot or another. You can see in all cases, this is an inset, where this inset is the full range of receptor snapshots, and the larger plot, larger axis, is just the first 40 snapshots. And basically you see a gradual improvement in the metric, so going up to what it's going to be it takes hundreds, or it takes about 100 if you just pick these snapshots completely randomly. And the blue and green, well, they're less random approaches. I'm not remember exactly what color is which, but one of them corresponds to if you pick them in order of the lowest to highest docking score, and the other in the order of lowest to highest binding potential mean force. Now, choosing the lowest to highest binding potential mean force is not a useful strategy, because if you have all the binding potential mean force calculations, there's no reason to pick a subset. But it is helpful to do a docking score, because you can quickly do a docking score against a few hundred snapshots and then just go into more intense calculations on a small subset of those snapshots. But yeah, if you choose based on those strategies, then you get to your final quality where when you have a lot of snapshots much more quickly. Actually, Pearson R, it's essentially no or very, very small number of snapshots are needed. But then to get the root mean square error down, it takes a few dozen. So we also have been considering how to use Markov models to get the binding free energy. As you probably are familiar with, these Markov state models are often used to piece together information from many short molecular dynamics trajectories and basically take advantage of the fact that when you do a molecular dynamics simulation, you might get a lot of local sampling, but not a lot of global sampling. But you can sort of piece together the transitions between local minima to get a global picture of what the energy landscape looks like. And if you, since Markov models have become increasingly used or widely used in, especially in protein folding, but it's also been used in terms of like looking for binding pockets on the surfaces of proteins, then yeah, if you have the sampling already, can we use them to get the binding free energy? That would be pretty beneficial, we thought. So we aim to demonstrate that you could do this with, again, our favorite model system, T for lysozyme. So first thing we did was we built a Markov model of T for lysozyme by doing 4 longish simulations from 4 different starting poses, and then using some off-the-shelf Markov model methods to pick out cluster centers get a free energy surface and look at the kinetics between them and like group them into similar or slow transitions between states. The important thing is, well, that we have the equilibrium probabilities of a bunch of different snapshots. We have the APO ensemble, basically, for this particular force field. So the first thing we tried is we said, okay, we have all these Markov model centers, Markov state model centers. We can use these centers and we can dock to them and get binding PMS from them. But it turned out in this particular system, a lot of these had very high binding potential mean force values. And we were a little stuck. Like we actually need low binding PMFs to get the binding free energy accurately. So we looked into this more. It looks like, okay, the binding pocket is collapsed. That's why it's always high. So we tried a couple of approaches over the last few years. We tried, okay, instead of just doing those number of snapshots we started, we just tried to increase the number of snapshots. We tried, okay, how about we minimize the receptor? And we do this with restraints, we do this without restraints, and just use this as an approximation. We were kind of hitting our heads against a wall. Eventually, though, Bing figured out that we can just say, okay, let's pick out the snapshots that have the largest pocket volume. And lo and behold, we get good correlation of Yank, correlation experiment, and with our other results based on the alchemical, where we took the snapshots of the protein from alchemical simulations. But this strategy wasn't, I didn't think it was representative of, like these snapshots aren't representative, they're just the biggest ones. So it might not work well in general. So we figured, okay, let's figure out different ways to pick out receptor snapshots. And we turned to some of our other work that is related to ensemble docking. Basically, the idea of this reduced ensembles is, well, you can have a full ensemble of snapshots from a simulation, but some of them are very similar to others. So we can cluster it and pick out ones that are representative, and we can do like a weighted average if we want to get the average docking score or average or the exponential average of the binding potential mean force to get the binding free energy. So yeah, you can pick out snapshots from the full ensemble in a random way or in a representative way. Like you can pick out something that's like the medoid of each cluster. And what I realized a few years ago is that when you do this in ensemble docking, this is an example of a statistical variance reduction technique known as stratified sampling. And stratified sampling is basically grouping your whole population into groups that have similar properties. So for example, if you wanted to know the average height in a room, you might say, let's get the average height of like the males and the females. And then we know the number of males and females, and then in the particular room, for example, and then you can calculate it that based on a weighted average of the number of the height based on male and female. Not a perfect example, but it's just an example of doing stratified sampling. And that's exactly what we're doing when we're doing this ensemble reduction. And you can use the same statistical tool to assess the quality of ensemble reduction, and that's referred to as the efficiency of stratification, or eta. And that's basically the ratio of the variance between stratified and simple random sampling. And for eta, based on this definition, it's better to have a lower eta. So we tried a bunch of different strategies to group together snapshots from several molecular dynamics simulations, and we basically identified some good strategies to pick out snapshots. And I won't go into tons of detail about this for time reasons, but our recommendation in this paper that you can read is to do hierarchical clustering based on the Jacquard distance between occupancy fingerprints. Occupancy fingerprints are basically a three-dimensional grid in the pocket, and if you have an overlap with the atom in the pocket, then it's 1, and if there's no overlap, then it's 0. And Jacquard distance is a way to compare these occupancy vector. Is basically a binary operation. So by applying this approach, we picked out 200 representative snapshots from 20,000, and then we can see what the binding potential mean force for these snapshots is. You can also compare it to the pocket volume. Turns out if you picked out the largest pocket volumes, you would get things that have a low binding potential mean force. And if you have very small pocket volumes, then you will get things that have very high potential mean force. But in between, you have a bunch of snapshots that the volume isn't tiny, but it's not huge either. And if we just pick the huge ones, we would be missing all this information here. So yeah, we did this. It works pretty well if you have 1,000 snapshots. And we're in the process of writing this up slowly since she's not working with me anymore. Okay, so we also have worked in the last few years on ways to apply the fast Fourier transform, which is common in molecular docking of proteins together, and also in the context of finding hotspot residues on proteins. And we showed that this approach can be used not only to determine interaction energies, but actually to determine binding free energies using implicit ligand theory. You can quickly calculate, using the fast Fourier transform, the interaction energy between the protein and its binding partner, or two binding partners, at a large variety of translations. So this is a 2D cross-section of, like, as you translate a benzene molecule across T for lysozyme, you see regions where you have near zero interaction energy. You have a lot of regions where, well, basically the maroon is infinite, or it's at this 18 or above. And then the tightest binding is within the pocket and also on the surface of the protein, according to the force field that we used. We can get a reasonable reproduction of Yank and AlgaeDoc results using these FFT-based estimates, but this is a one-step perturbation that's essentially being used to reproduce the alchemical transition, and it's not a perfect reproduction. And we also have shown in this paper, and also a paper that just came out, that you can use not only the binding PMFs to estimate an absolute binding free energy, but you can compare the binding free energy between a ligand and another ligand if you do a simulation of the protein bound to a reference ligand. So this is described in more detail in the paper, but this approach is, I anticipate, will be useful if you have similar hollow ensembles, or ligands with a similar hollow ensemble. So if you have like a nuclear hormone receptor that whenever it's activated, where all agonists have a similar mechanism, perhaps, then you might be able to identify what's the agonist. So yeah, we thought about this idea in the context of the estrogen receptor. So basically, here's a crystal structure of estrogen receptor alpha, and it's known to have an active and inactive form, although there's a little bit of, well, there's a little controversy. People will say that this helix is in different positions in the inactive form, and this helix alpha, helix 12, is always in this position for active ligands bound to agonists. People have tried to dock to these and sort of say, okay, can we identify what is active and what is inactive? And that doesn't work. There's a very poor performance of figuring out what's an agonist and what's an antagonist. So what we did, we tried to calculate binding PMFs. We also did interaction energies using the same force field, basically the GBSA. And we said, okay, these are the agonists against the active crystal structure. Here are antagonists versus the active crystal structure. And you can see that the antagonists actually have a very low interaction energy. They actually are on average lower than the agonists. Like you can see the position on the y-axis. But if you look on the x-axis, their free energies are not as low as with the agonists. So some of the, I guess what some of the dogma in this area has been that your antagonists just don't simply don't fit into the pocket, but that's not true. They fit. They lose a lot of entropy when they fit, opposed to the agonists. They don't lose as much entropy. So this is a preliminary result we're hoping to getting around to publishing in the near future. These are the students. Well, this is a couple of years ago now at a free energy conference in Boston. There was a bit of a lull in my group for a bit, but now I have 5 PhD students who just came in this year. So hopefully we'll be pushing this forward pretty soon. And thank you for your attention, and I'm happy to answer any other questions that you have.
Okay, thank you very much, David. That was very nice. I learned a lot myself. Again, I mean, there is always more to learn about free energy and how you calculate it better and more accurately. Thanks. So I asked a lot of questions. So now I still have a couple of more questions that I can ask maybe before people warm up and ask it. But please, if you have any questions, you can just actually turn on your microphone and start asking your question. So let me start. First of all, the assumption, if I understand, is that the presence of the ligand is not going to affect the energy landscape of the apoprotein. Is that an assumption for the implicit ligand theory, or it's not true, what I said? So like if you simulate the protein in the absence of the ligand, the apo ensemble, are you assuming that this ensemble will be the same as the ensemble in the presence of the ligand once the ligand binds?
No, we don't make that assumption. We basically account for the differences between the ensembles. We basically, you can say that the binding potential amine force is a reweighting factor. I didn't go into that in detail, but it's basically a reweighting from the apo to hollow ensemble.
I see. Is that what you do? Kind of to some degree, you even address this more when you are taking your active snapshot. No. That was actually about the ligand. Never mind. So
right.
Yeah.
There were a couple of ways that we addressed the discrepancy between the apo and the hollow ensemble. Yeah, basically, if you just pick up the apo ensemble for T4 lysozyme, in many cases, the force field predicts that the pocket is collapsed. So those pocket collapsed snapshots are not useful in the hollow ensemble. So we sort of do this selection of snapshots based on the binding site properties, so that we make sure to include snapshots that are relevant to the hollow ensemble. Another approach that we've taken is that we've said, let's just do simulations with ligands in them and then reweight it to an apo ensemble. That's what we did in the first paper, essentially. But we can also, it is possible just to do a simulation with a ligand and then take those snapshots, remove the ligand, and get the relative free energy of a different ligand to it. So yeah, at least with T4 lysozyme, it's not very it's not very flexible, but it has enough flexibility that you can't just pull apo snapshots and hope to get an accurate free energy for a small molecule. And that's what some of the challenges we dealt with getting this method to work in a practical sense. I mean, back in 2012, I knew that it worked in a theoretical sense. The question remains still is how good is it for practical purposes.
Cool. Jim Tufts, I promoted you to a panelist. You can actually turn on your microphone and ask your question.
Hi. So I'll just full disclosure, I'm one of David's new PhD students. But I wanted to ask a question. Is it really unfair to bias the snapshots to have to be large enough to actually fit the ligand in the pocket? Because if you're docking against rigid receptors, isn't it possible for a conformation change to occur or be favorable if the binding event is, energetically favorable? So I guess my point is it really unfair to make sure that there's enough, or at least some that are big enough to fit the ligand, or unrepresentative, really?
I'm not sure what you mean by unfair.
Unrepresentative, I guess.
Yeah, I mean, it's not going to be it's not going to be that necessarily that prevalent in the APO ensemble, but basically any configuration should be accessible, and what we need are snapshots that are relevant to the hollow ensemble, and there are different approaches to getting that. As far as being fair or unfair, I mean, we have biases that we introduce, and then we basically have to account for those biases.
Okay. Yeah, I guess the question's more just about if you can never access those biased states, or the states where it's big enough, let's say just due to your stratification, won't you just miss things as well? So I feel like it's necessary to at least have some that are big enough, but you'd have to maybe weight it somehow to have it be more representative of, the bound ensemble. Does that make sense?
Well, I mean, I was saying the binding PMF is the reweighting from the APO to hollow ensemble. But yeah, there's a chance that if you don't simulate enough, that you'll miss the bounds, the configurations of the protein that are relevant to the bound ensemble. And if you do that, then you just get a high binding affinity or binding free energy, and it's not going to be predicted to be, it's just going to be biased high. And that's also true if you do a free energy calculation with a flexible receptor. Like you just might miss a conformation of the flexible receptor that accommodates the ligand. So there's no unique deficiency in this approach, other than you might do a simulation. If you do a simulation without the ligand, then you're less likely to get conformations that are relevant to when the ligand is bound. I did not plant that question.
Thank you.
Imad, may I ask a general thing?
Please.
Of course, don't understand anything. I'm retired for 20 years. But I'm interested in binding in plant proteins, and you have done 85 different proteins, I heard. Is there any interest in going into, let's say, photosynthetic proteins like photosystem 2, which has been crystallized, and the extra structures available? And I have especially interest in a ligand called bicarbonate, which is bound to one of the proteins in the water oxidase system. And is there any way that anyone in your field can get interested in working on that? So it's not a question on your research seminar, which was exciting, but not understandable to me, being not in the field of theory. So could you help me with that? Or could I write to you and you help me figure out how these systems work? I mean, there are a lot of people who are in it, but nothing has come forward to me that I can feel is going.
So I can give you a short answer, Govinji. We either need a company who is interested in that, or we need a grant who supports it. That's usually the major bottlenecks for
yeah, the agricultural company, the companies which deal with agriculture might have some interest. So I have to convince them, I guess, first, is it? But it's a scientific question, so,
I mean, David's method can be applied to essentially any ligand protein interaction.
Yeah, right.
There is no restriction or limitation to apply that to your problem, I guess. But again, I mean, David, you can.
Yeah. What does David say?
Well, yeah, I mean, I echo what Imad says.
Yeah. I get the same.
Not knowing specifically what the question that you're.
Yeah, well, I can understand the questions. I have the specific questions. The question is, would anybody be willing to do it without money? Because I have no money.
I guess the answer is probably no, Ravinji.
I see.
If you don't have the money, that's usually the sort of a limiting factor. But
Okay,
actually, I have another question, David. So, have you considered explicit water? Like we know that in ligand binding, even for docking or free energy calculation, sometimes there are water molecules that stay in the binding pocket, right?
Right.
So, and if your method is fast enough, I can imagine you can do one round without water and then identify maybe empty pockets around the ligand that might stay hydrated. And they do another round of ligand binding calculations with water's molecules in there. Have you thought about water molecules and tried to do something about them? No.
Well, I've thought about it, but I haven't tried to do anything about it. So you can consider a water molecule as part of the ligand. That's one way to do it. Or sorry, part of the receptor.
Yeah.
Or you can think about it as like a second or third ligand, and you can do like a cooperative binding free energy calculation. So yeah, actually talk about the theory of including it as a part of the receptor in the original paper on implicit ligand theory, but just haven't gotten around to like doing anything that demonstrates that yet.
I mean, if it takes 1 or 2 days on one CPU for you to get to sort of probe one conformation of your receptor, so you can try with water, maybe with 2 waters or with water at different positions, and that might be interesting. Anyway, thank you. So any other questions, guys? Again, you can just go ahead and ask your questions if you have any questions. If not, I would like to thank David one more time. So David, we're going to have a kind of another Zoom session. You have that link?
Yes, I do.
So we're going to see you in the next Zoom session soon. I'm going to talk with you maybe for 5 minutes privately, and then some of the group members are joining to show you some of their results and have your feedback. Thanks again for the seminar. Thanks for everybody to participate and join, and have a great evening. See you, David.
Thank you for inviting me again.